Hello viewers. In today's lecture, I will be discussing about DNA packaging with special reference to nucleosome assembly. But before I begin, I would like to mention that this presentation is a part of the initiative taken up by the Zoological Society of Assam to reach out to the students, particularly those who are pursuing their graduation in zoology. So let us begin today's lecture and try to understand the very concept of chromatin, DNA packaging, and the importance of nucleosome assembly. The learning outcome or objectives of today's lecture is to understand the concept of chromatin and the different levels of chromatin organization. So this will include understanding the primary level of chromatin organization, that is the beads on a string fiber, understanding the secondary level of chromatin organization, that is the 30 nanometer fiber, and understanding the tertiary level of chromatin organization, that is the formation of mitotic chromosomes. We will also try to understand the basic structure and the importance of nucleosome assembly in DNA packaging. So with these objectives in mind, let us begin today's discussion. Let us quickly recall the differences in the genome size of prokaryotes and eukaryotes. The eukaryotic genome is much larger than the prokaryotic genome. If we consider the haploid human genome, it has a size of 3,200 million base pairs, in contrast to that of the bacteria E. coli, which is a typical prokaryote and has a genome size of 4.6 million base pairs. If we recall the structure of the DNA, which has a diameter of 2 nanometer, each base pair in the Watson-Crick model of DNA is around 0.34 nanometer long. Now if we do a mental calculation, the DNA as such will be about 2 meters long, and it is practically impossible to fit a DNA of this length into a membrane-bound organelle called the nucleus inside the eukaryotic cell, which has a size of only 10 to 100 micrometer. Here comes the necessity of DNA packaging or DNA compaction. The DNA does not lie naked inside the cell. Instead, the helical structures of the DNA forms complexes with certain basic proteins called histones, and we will be discussing about these in more detail subsequently in the presentation. This leads to the DNA to be neatly packed into chromosomes. If we consider the cells of the human body, most cells of the human body are diploid with 23 pairs of chromosomes. Now what does this mean? This means that the DNA in each cell is neatly packed into these 23 pairs of chromosomes. It is fascinating to understand how remarkably the DNA is tightly packed to fit inside the nucleus, which has a diameter of only 5 to 10 micrometer in every eukaryotic cell. This is accomplished by the formation of a fibrous complex of eukaryotic DNA and histone proteins and is referred to as the chromatin. The basic structural unit of organization in the chromatin of eukaryotic cells is called the nucleosome, which consists of a negatively charged DNA wrapped around a positively charged histone core. That is, the primary protein component of chromatin are histones, which anchors the DNA. And the primary function of chromatin is to package DNA into a more compact and denser shape. Before moving further into understanding the nucleosome assembly and the different levels of chromatin organization, we need to have a clear idea about the types and properties of histone proteins. Histones are small basic proteins which are found in the chromatin of all eukaryotic cells. The histone proteins have molecular weights between 11,000 to 21,000 Dalton and are rich in basic amino acids arginine and lysine. So this is a typical characteristic of histone proteins. All eukaryotic cells have 5 major classes of histones differing in molecular weight and amino acid composition. These are H1, H2A, H2B, H3 and H4. The histones H2A, H2B, H3 and H4 are referred to as the core histones because they are responsible for making up the nucleosome core, whereas the histone H1 is referred to as the linker histone because it binds to the linker DNA. We will be discussing about these in more detail subsequently. With these basics in hand, let us now try to understand the nucleosome and its assembly. The nucleosomes are the building blocks of chromosomes, and it was first observed as particles or spheroid chromatin units in the electron microscope by Don and Ada Ollins in 1974 at Oak Ridge National Laboratory, Tennessee, USA. The nucleosomes were later described by Roger Kornberg in 1974 while he was working at the MRC Laboratory of Molecular Biology in England. Kornberg described that the nucleosome consisted of histones and a 200 base pair segment of DNA. The crystal structure of the nucleosome was later deduced, and it was found that the nucleosome consisted of a histone core wrapped with 146 base pairs of superhelical DNA. Additionally, the left-handed DNA superhelix wraps around the histone core in 1.67 turns. The histone H1 binds to the linker DNA and plays a very important role in DNA packaging. The nucleosome consists of approximately equal masses of DNA and histones. So the predicted mass of a nucleosome that contains the histone H1 is around 262 kilodalton. If we look into the shape of the nucleosome, it corresponds to a flat disc or cylinder of diameter 11 nanometer and height of 6 nanometer. Another important point is that the N-terminal tails or the N-terminal ends of these histones not only provide a guide for the DNA strand to wrap around those histones, but can also undergo modifications such as acetylation, methylation and phosphorylation. These reversible modifications can influence how tightly the histones bind to the DNA, thereby affecting the expression of specific genes via chromatin remodeling. At this point, I would also like to mention that the strong association between histones and DNA is attributed to the very nature of these molecules. Histone is largely made up of positive amino acids like lysine and arginine, which make the overall structure positive. DNA, on the other hand, is negatively charged due to the phosphate groups in its phosphate sugar backbone. Because of these factors, the histones can bind with DNA very tightly via hydrogen bonding. Let us now look into the assembly of the histone core in more details. The nucleosome core particle or the histone core contains two copies of each histone protein, H2A, H2B, H3 and H4. The assembly of a nucleosome is initiated by the formation of a H3H4 tetramer. So basically, the association of this H3H4 tetramer is the first step in nucleosome assembly. This tetramer then binds to a double-stranded DNA and recruits two H2A, H2B heterodimers to complete the histone core. The crystal structure of the nucleosome core particle was first deduced by Tim Richmond and his research group in 1984. As you can see in the image on the extreme left, the DNA is wrapped around the histone core. It is important to note that the H3H4 tetramer binds to the middle and ends of the DNA wound around the histone core, whereas the rest of the DNA in the histone core is bound by the H2A, H2B dimer of the histone core. The nucleosome thus provides the first level of chromatin organization, compacting the DNA about 6-fold over the length of naked DNA, resulting in a beads on a string fiber of approximately 10 nanometer in diameter. Herein, the beads represent the nucleosome, whereas the string represents the linker DNA. In totality, the spacing of the nucleosome beads provides a repeating unit of about 200 base pairs. So this statement was experimentally proved by Roger Kornberg in 1974. In one of his experiments, partial digestion of chromatin with micrococcal nuclease, which is an enzyme that degrades DNA, was found to yield DNA fragments of approximately 200 base pairs long. In contrast, a similar digestion of naked DNA, that is DNA which is not associated with any kind of protein, yielded a continuous smear of randomly sized fragments. These results suggested that the binding of protein to DNA in chromatin protects region of the DNA from nuclease digestion, so that the enzyme can attack the DNA only at sites that are separated by approximately 200 base pairs. So basically, nucleosomes, that is the basic structural unit of chromatin, are repeating units of about 200 base pairs. As of now, we have understood the assembly of the histone core, which is made up of the 4 histone proteins H2A, H2B, H3 and H4. But what is the role of H1 histone? So we know that adjacent nucleosomes are connected by short stretches of DNA called the linker DNA. We need to understand the fact that the secondary level of chromatin organization involves interaction between nucleosomes of the 10 nanometer fiber, leading to a more condensed chromatin fiber called the 30 nanometer fiber. This packing requires one molecule of histone H1 per nucleosome core. In other words, the binding of H1 histone to the linker DNA stimulates the formation of the 30 nanometer fiber by pulling adjacent nucleosomes into close proximity. It therefore plays a very important role in the secondary level of chromatin organization. Let us now look into the structure of the 30 nanometer fiber, which is the secondary level of chromatin organization. Binding of the H1 histone to the linker DNA leads to further condensation of the chromatin by coiling into a 30 nanometer fiber containing about 6 to 8 nucleosomes per turn. Though the structure of the 30 nanometer fiber has not been fully determined, and it is still a matter of debate whether it is a dominant feature of the chromatin, but it is suggested that the structure of the 30 nanometer fiber varies between species based on the length of the linker DNA to which the H1 histone binds. There are two hypothesized models for the organization of the 30 nanometer fiber. The solenoid model, also known as the one-start helix model, and the zigzag model, which is also known as the two-start helix model. In one-start solenoid model, consecutive nucleosomes interact with each other and form a helix with bent linker DNA connecting each pair of nucleosome cores which follow each other immediately along the same helical path. In other words, in this particular model, the nucleosomes are stacked on top of each other, forming a helix, and the linker DNA is hidden on the inside of the helical core. In the two-start zigzag model, it is basically of two types: the helical ribbon model and the crossed linker model. In both these zigzag models, two rows of nucleosomes form the zigzag model, and the straight linker DNA, in contrast to your bent linker DNA as in case of the solenoid model, connects alternate nucleosomes that are either cross-linked as in case of the cross-linker model or helically arranged as in case of the helical ribbon model. In other words, in the zigzag model, the nucleosomes are arranged in a zigzag manner. This form actually requires a longer linker DNA as compared to the solenoid model, and therefore it is likely to be observed in species with longer linker DNA. Overall, the 30 nanometer fiber provides an approximately 100-fold compaction of DNA. The final tertiary level of chromatin organization requires the further folding and compacting of chromatin fibers into the three-dimensional structures of interphase chromatin or mitotic chromosomes. In order to achieve this, the 30 nanometer fiber folds into large loops which are held together by nuclear scaffolding proteins at the base of each loop. The looped fibers further coil again to form the condensed chromosome in order to achieve a 10,000-fold compaction. So these three levels of chromatin organization ultimately leads to the packaging of DNA inside the nucleus of a eukaryotic cell. In addition to this, I would also like to mention that chromosomes are not always present. They form when the cells are dividing. In other times, the DNA becomes less highly organized and remain as chromatin fibers. Based on the degree of compaction of packing, the chromatin fibers are classified as heterochromatin or euchromatin. The tightly packed regions of the chromatin fibers is known as heterochromatin, whereas the regions which is loosely packed are known as euchromatin. However, the structural and functional features of heterochromatin and euchromatin will be discussed in a separate presentation. So far, we have discussed about DNA packaging in eukaryotes. But what about DNA packaging in prokaryotes? Is it similar or is it different? It needs to be pointed out that histone proteins are absent in prokaryotes. That is, there is no nucleosome assembly in prokaryotes. However, the bacterial DNA is also highly organized and compacted into the nucleus. Certain histone-like proteins such as HU protein, which is a 2 subunit protein with a molecular mass of 19,000 Dalton, is present abundantly in E. coli. The bacterial chromosome also seems to be much more dynamic and irregular in structure in comparison to the eukaryotic chromatin. So these are the books and research and review articles that has been referred for preparation of this particular presentation and may be referred for deeper understanding of the subject. Drop in your queries and feedback at the email address given below, and if you found this video helpful, like and subscribe to our channel. Thank you.